All right, so we're going to go over the second part of the building blocks of life. So last lecture we went over carbohydrates and lipids. Now we're going to start by going over proteins and nucleic acids. All right, I apologize. I think this is going to be a little lecture heavy today, but I also think we might have a chance of getting out here a little early, so we'll see what happens. All right, so you guys should have watched a video about amino acid structure. So I'm not going to spend too much time on this. I do expect you guys to, of all the molecules we're talking about, be able to draw this molecule, all right? Because this is pretty important. All right, amino acids are amazing. They're awesome. They're part of so many important things. It's not too complicated. I want you not only to be able to draw it, but know which groups are what. All right, so the carboxyl group, the amino group, where the hydrogen is, and where that R group is. All right. Now I don't expect you guys to know every single R group. I think that's a little tedious, and honestly, I don't have that memorized, so I'm not expecting you to guys have that memorized. So R groups, just know it's an R group. All right. We know that there's 20 different of these amino acids, depending on what R group is attached. All right. So many different side chains that can be attached, all right? They can be branching, they can be linear, they can be in these rings. Have many different side chains that make up these 20 amino acids. All right, I'm going to kind of go fast because like I said, you watched a video on this, you should have kind of an understanding of it. So that moves us into proteins. All right, so proteins, they're involved in almost every bodily function that we have, all right? Proteins are so important. You're going to see this come up time and time again in my lecture today. They're also very diverse, all right? So proteins, they're made up of just 20 amino acids, all right? There's just 20 basic building blocks to a protein, all right? But because they are put together in different ways and their structures are different, they can have vastly different functions, right? So much so that there are, thousands upon thousands of different proteins, all made up from just those 20 amino acids, all right? They're also very highly selective, all right? So a protein probably only has one or very few jobs to do, and that protein is only going to do that job, all right? They are highly selective in what they do because their structure is so specific to their function. All right, so they're made up of 20 amino acid monomers. All right, so if we put those amino acids together, we're getting a polymer, all right? And when we talk about amino acid polymers, we refer to these as polypeptides. So those are molecules of amino acids joined together by peptide bonds. All right. Going to try to slow myself down here. I think I'm still a little amped up from lab. So I'll give you guys just a moment to jot this down, but remember that this is all available for you, all right? On the slide, you can write this down later if you need to. So polypeptides, molecules of amino acids joined by peptide bonds, they're always going to have an N terminus on one end and a C terminus on the other end. So let's look at this. All right, so let's break this picture down into smaller bits here. So first, let's look at the formation of a peptide bond. All right, so a peptide bond, that is a covalent bond. And we know in covalent bonds, there's a sharing of electrons going on. All right, and this is a bond. This covalent bond is formed by a dehydration reaction. All right, so we've seen this many times now. We are taking an oxygen from the carboxyl group, 2 hydrogens from the amino groups. We're synthesizing a water molecule, which is a byproduct here, and forming this peptide bond in the meantime. All right, so dehydration reaction, covalent peptide bond. It's a bond we call 2 different amino acids joined together. All right, now I said there is an N terminus and a C terminus. All right, so the N terminus is going to be the free amino group. All right, so we're joining all of these together. Most of these amino acids, both their amino group and their carboxyl group, are going to be part of these bonds. All right, they're forming these bonds. But on either end, right, those open ends, we're going to have a free amino group and a free carboxyl group. And the terminology for this is just to refer to this as the N-terminus, right? You can think about that because it has a nitrogen in it. And the C-terminus. Again, you can help remember that because there's a carbon there, all right? Let's see here. I'm just going to briefly go over how we're going to number them. We're really not going to use this system, but because I put this image on here, I want you to be able to understand the image. So we can label each amino acid in the sequence, right? And this is just a very simple sequence here of only 8 amino acids. And they just labeled it from the N-terminus to the C-terminus, 1, 2, 3, 4, 5, all right? Now scientists can do this for amino acids sequences, these polypeptides that are hundreds to thousands of amino acids long. All right, so this is a very, very simplified version. All right, what do proteins do? They do almost everything in our body. Anything that you can think of, there is probably a protein involved. All right, so very, very diverse. I'm just going to quickly go through this list here. So they can be used as enzymes, speed up reactions. All right, next lab is going to be a lab on enzymes. So we're going to be looking at a protein. They can be used in defense, such as like antibodies, attacking pathogens. They can be used in your muscles to help with movement, so motor and contractile proteins. They can convey signals between cells. They can provide structure. And they can also transport materials. Maybe they're transporting larger molecules between the phospholipid bilayer of a cell. All right, they have so many functions because they are so important. All right, so they are so important that I made this its only slide. Proteins are important. All right, so what makes them important is they have so many different functions. And that function is all due to the structure of their protein, how they are made. All right, so not only are they diverse in size and shape, all right, so the number of amino acids they physically contain, but they're diverse in the chemical properties of their amino acids as well. All right, so not every protein is going to have the same sequence of amino acids. That amino acid sequence is going to change around. All right, so they're very diverse. Let's see here. So protein structure. Structure equals function. That's something you're going to see so many times throughout this lab. This lecture. So let's talk about the protein. A protein is going to be one or more polypeptides that have folded or twisted into some three-dimensional shape. All right, so it's more than just a line of amino acids. It's these amino acids that have been twisted, they have been folded up, they have created some sort of unique shape that is specific to their function. Now, these proteins, besides having amino acids, they can also have some carbohydrates or lipids attached to them that might help them aid in having a very specific function to do a specific job. All right, so they can have other molecules attached. And I threw this picture up here of a lysosome. This is an enzyme. And when you look at the structure of proteins, maybe you've seen pictures kind of like this one on the top. It looks like kind of like this ribbon floating through the air. Maybe you've seen this one that's more chunky. All right, they're just two different depictions of the three-dimensional structure of this enzyme. But we can see that in both of them, they have this groove, which for this enzyme, this is important to how it functions. All right, so I've said structure equals functions, function many, many times here. So how is this structure created in a protein? All right, so protein has 4 levels of structure. All right, they have primary, secondary, tertiary, which is this one here, and quaternary. Now we're going to break these all down and talk about them individually here. So primary, this is the basic structure of a protein. All right, you can't get more basic than this. This is simply the amino acid sequence. All right, so what amino acids are in the specific sequence of the polypeptide? All right, and again, these amino acids, they're different only by their R groups, only by those side chains. Now this is going to be encoded directly by our genes. All right, this is embedded in our DNA and how to produce these amino acid sequences. All right, the secondary structure, this is where we get some fun things going in with our structure here. So in secondary structure, we can have chemical and physical interactions that cause some sort of folding. All right, so that folding can be irregular or they can be very structured. So we're going to talk about these two main structures of secondary structure called alpha helices and beta pleated sheets. All right, these are kind of the two determinants of a protein's characteristics that they will, structure equals function here. All right, so let's talk about the alpha helix. So the alpha helix is this one here, right on top. All right, these are going to be stabilized by hydrogen bonds that are at regular intervals. I don't think this picture does it justice, so I'm going to jump back to the one you've already seen, kind of zoomed in here. All right, so they have these hydrogen bonds that are binding this backbone together. Now, when I talk about the backbone, I'm talking about the amino group, that carbon, the hydrogen, and their carboxyl group. I'm not talking about the side chain. I'm not talking about the R group, right? So that backbone is everything besides the R group. And these hydrogen bonds are bonding this backbone together. All right, now notice in the alpha helix, it's just this one polypeptide that's being hydrogen bonded to itself, essentially. All right, one strand. Now in the beta pleated sheets, all right, we have kind of the same thing going on. All right, we're still talking about the backbone, and we're still talking about hydrogen bonds. In fact, those hydrogen bonds are essentially in the same place. All right, but now we have two strands of this polypeptide that are parallel to each other. All right, so this one it's kind of folding in upon itself. This one, the two strands are parallel to each other. All right, so that causes a totally different shape to occur, and it causes this beta pleated sheet. All right, so then besides these very structured sections, we also have these random coiled regions. All right, they seemingly have no pattern to them. They look very random. They're anything but random, however. So however they have decided to fold up, that is directly going to relate to its function. All right, we just call them random because they're not nice and neat like these alpha helices and beta pleated sheets. That makes sense in the secondary structure. All right, so next layer is the tertiary structure. All right, so this is going to give the protein kind of its more complex shape. All right, and this is the final level structure for a single polypeptide chain. All right, so a single polypeptide chain can't be more complex than the tertiary structure. All right, so in the tertiary structure, it basically folds and refolds upon itself. And this is where those side chains start to interact to help with the folding. All right, so the hydrogen bonds, they help with the secondary structure. These side chains, they're helping with the tertiary structure. Now notice this is one polypeptide, all right? So we have our strand of polypeptide going in a different direction here, causing those beta pleated sheets. We have a single strand, hydrogen bonded to itself, causing those helices, right? But this is one polypeptide. Now if we add more than one polypeptide together, this is where we can get a quaternary structure. All right, so this is going to be two or more polypeptides. We often refer to proteins that have two or more as having individual protein subunits. All right, so the subunits would be the individual polypeptide chains. Now we can have these proteins be either the same type of protein over and over and over. They could be, the subunit could be the same polypeptide, all right, folded in the same way. Or it could be multimeric, which can be composed of different type of polypeptides. All right, so if we look at this one on the bottom here, I threw this image up here because if you have not talked about it already, you probably will talk about it in lab. This is hemoglobin. All right, so we're talking about sickle cell disease in lab. And so the hemoglobin is composed of 4 subunits. All right, it has an alpha globin and a beta globin. right? Those are the 2 subunits, and each of those are in there twice, right? So it has these 4 subunits. So I would consider this one to be multimeric. Even though it might be just a slight difference that makes it an alpha versus a beta, still a different protein. All right, so there are different factors that can promote this folding and promote the stability of the protein. Now we've talked about the majority of these already, right? Hydrogen bonding, right? We've seen that with secondary structure. Ionic bonds and other polar interactions can help determine their structure as well as hydrophobic effects, so what part of their side chain might be water-fearing, might determine how they bond to another part of the polypeptide chain. But we also have these 2 other effects that we haven't really covered yet in class. The first is the van der Waals forces. All right, so van der Waals forces are atoms within molecules will have a temporary attraction to one another if they're an optimal distance apart. All right, so it's these temporary attractions between different molecules. We call these van der Waals forces. This helps determine its folding as well. I'm not going to go into any more complexity than what I just stated there. I'll restate it there, though, for those taking notes, because I did not write this out on the lab here, or on the lecture. Still in that lab mode. I keep saying lab. So van der Waals forces. Atoms within molecules having temporary attraction to one another if they're an optimal distance apart. All right, so that would be the van der Waals forces. Next, we have the disulfide bridge, right? This is another type of bond that can help promote folding. I actually threw in another slide here, which I don't think is in your copy of the slides, but I thought an image might help you understand this a little bit better. So on a disulfide bridge, this is where we have an amino acid called cyteines, all right? They have sulfur in them, all right? And they can bind to one another via their sulfhydryl group, all right? So this is very specific to this one amino acid, and they have the ability to bind to each other via those sulfhydryl groups, all right? And the resulting bond is called a disulfide bridge. Any questions so far? All right. So let's briefly break apart, do a group question here. If you've had lab, this could be an easier question. All right, so talk amongst yourselves, come up with an answer, and we will come back.
Oh, yeah, yeah.
For the soccer season, is that the
Football.
football?
Yes, I do.
says. All right, the volume has considerably decreased. So I'm assuming that means everyone has had a chance to think about this and talk about it with one another. Who would like to answer this? Would it affect the tertiary structure?
We said yes because if you are changing up the primary structure, you'd be changing up which amino acids are in which places, and thus I changed like how many hydrogens you have and like hydrogen bonds, as well as like van der Waals, and you said that's determined by the atoms that are in the amino acids.
Yeah, exactly. So if you've had lab, maybe you kind of had an idea when we were talking about sickle cell disease. All right, so sickle cell disease, we're changing up just one amino acid, all right, and that is affecting the entire shape of the protein, all right, the protein hemoglobin. All right, so even if we change something at the very basic primary level, because we're changing the molecular makeup, it's going to change the amount of bonds it can have. It can have, if it can have bonds at all, maybe it can have a different type of bond. All right, that's all going to influence how it's folded together. And that in turn is going to affect its overall shape and overall function. All right, so if you haven't had lab yet, maybe that's a good thing to ponder, and I'll maybe give you an edge up in those next labs coming up here. All right, so in this here, sickle cell disease, I think there's like 600 amino acids, and we've only changed one, right? And just a change of one single amino acid has caused this dramatic change in shape of the overall red blood cell. All right, so pretty cool. All right, so because we have this shape equals functions, all right, we can have new properties emerge from complex interactions between different proteins, all right? So primary sequence of a protein determines the 3D structure, all right? And we know that if we change the primary sequence, it can totally change the 3D structure, but that also could change its ability to interact with other proteins. And I threw this picture up here. It's also in your book, but you can see how specific these are shaped and how it's going to fit in its complementary proteins so well. So imagine if that shape was changed and how specific this isn't going to function very well anymore, all right, if we had some sort of mutation. All right, so shape equals functions, proteins super important. Any mutation just in one singular amino acid can have drastic results. All right, kind of just went over this. So if it loses its shape, though, it won't function. All right, maybe it can't function with its complementary protein, can't fit inside the grooves as well. We can denature a protein. All right, we can make it lose its shape. This can be done a variety of ways. So if the pH level is too acidic, too basic, we can denature it, make it unfold. Salt can also interrupt those bonds that help the folding interactions within a protein. And also temperature, which I believe we're going to look at next week in lab too, is temperature and enzymes. All right, so a common thing is if you raise the temperature of a solution that contains an enzyme or contains a protein, it's going to cause it to unfold. Right? Now what that threshold is for different enzymes might vary, all right, but it gets to a point where those bonds, they are not going to be stable anymore. They're going to start to break apart. We're going to get unfolding or a denatured protein. All right, any questions about amino acids and proteins? All right, let's move on. Talk about nucleic acids. All right, nucleic acids. When we're talking about nucleic acids, we're talking about DNA and we're talking about RNA. So, DNA, deoxyribonucleic acid. All right, this is always going to be double-stranded. It's always going to stay in the nucleus, and it's going to store genetic information. All right, that's probably pretty basic for you guys. Everyone has seen the double helixes of the DNA strands. We also have RNA, which is a ribonucleic acid. Right? RNA is typically single-stranded. It can leave the nucleus, and the RNA basically decodes the information stored in the DNA and decodes it into protein-building instructions. We're not going to get to that now. That's going to be a few weeks down the road. We'll start talking about that. All right, but RNA, DNA, they're made up of the same type of monomers, nucleic acids, also called nucleotides here. So the monomers, nucleotides, are made up of the same thing. They have different functions. Let's start by talking about the nucleotide. All right, so the nucleotide. This is a molecule. I expect you to know what the different groups are that make up a nucleotide. So the first group is going to be a phosphate group. All right, it has this phosphorus atom in it. It also has a 5-carbon sugar. All right, it's either going to be ribose or deoxyribose, depending if we're looking at RNA or DNA. And then it's going to have a single or a double ring of carbon. All right, and these are known as bases. All right, now in this image, it can be kind of confusing because it looks like they have 2 phosphate groups. They're just showing that you can link them together via their phosphates. All right, so this would be the start of a new molecule. All right, so 1 monomer, 1 nucleotide, phosphate, carbon ring, or 5 carbon, and either a double or single ring of carbon as their base. All right, now they're linked together in a polymer by their sugar phosphate backbone. Right? So those phosphates play a key role in linking all the nucleotides together. All right, so actually let me back up because I want to mention how it's numbered in the sugar. All right, so the carbons in the sugar molecule are going to be numbered, and you'll often see this when learning about DNA. All right, so there's the 1, 2, 3, 4, 5. All right, and the phosphate can bind the different nucleotides together by binding the sugars together between the 3 and the 5 carbon. All right, and we'll see this a little in depth in I think next slide or the next slide after. All right, so a little bit more in depth of nucleotides here. All right, so we have our phosphate group, we have our 5-carbon sugar, and we have a base. Now this 5-carbon sugar can be ribose, can be deoxyribose. Notice that they're very similar, right? They have just a change in 1 atom in where it's positioned, but that causes a totally different molecule, all right? So we have our 5-carbon sugar. We also have our nitrogenous base. All right, so our bases can be split into 2 main groups depending on how many carbon rings they have. They can either be pyrimidines or purines, right? These pyrimidines is cytosine, uracil if we're talking about RNA, and thymine if we're talking about DNA. All right, they have a single ring, and there's 3 of them, all right? 2 of which can only exist in either RNA or DNA. Now we also have the purines. Now the purines are the double carbon rings, right? That's guanine and that's adenine. All right, so I expect you guys to not necessarily look at a molecule and tell me, oh, that's a guanine, easy. Nah, I'm not going to expect that level, but I will expect you to know what occurs in RNA, what occurs in DNA, what the 5 bases are, and what category they fit in. All right, are they a purine? All right, so a DNA strand here. All right, so this is where we can kind of see where the phosphates play an important role in creating that backbone. So right now we're just looking at a single strand. All right, we're not looking at the whole helix yet. So the phosphate group, again, is linking that 3 carbon to that 5 carbon. All right, so we're forming this backbone. So whenever we talk about backbone, we're talking about this phosphate backbone. All right, I also just kind of put this image up here because I think it really illustrates different types of bases. I think it's just an overall good image. Let's move on to what kind of bonds are formed to create that backbone. All right, so we have our phosphate group. It's being linked between the 3 and the 5 carbon. All right, this bond is being formed. All right, so the individual bond between the phosphate and the 3 and the phosphate and the 5 carbon, those individual bonds are called ester bonds. All right, and they are formed via dehydration reactions. All right, so we're taking away water and covalently bonding our phosphate to either a 3 or the 5 carbon. All right, so we have water as a byproduct. Now together, these 2 ester bonds forms a phosphodiester linkage. All right, often called a phosphodiester bond. All right, so that's the ester bond on the 3, the ester bond with the 5. Together, we call that a phosphodiester linkage. A linkage because phosphates in the middle, right? And they're linking the two nucleic acids together. All right, so we've all seen the structure of DNA. We've seen the double helix. What plays a crucial role in this structure is hydrogen bonds, all right? We are using hydrogen bonds to create this helix. And we know that the hydrogen bonds are always going to form between very specific base pairs, all right? A is always going to be bonded to T, so adenine to thymine, all right? Or guanine to cytosine, right? They're always going to form like that. We're always forming a purine to a pyrimidine, right? They're very specific, and we can see that here. And then this is going to help create that shape, and exactly where those bonds are positioned is helping kind of create this staircase shape that kind of wraps around and creating this double helix. All right, I'm not going to go into too much more detail about how that's formed because we're starting to talk a lot about physics and very complex individual things about the molecules or the atoms within each molecule here. All right, but essentially these hydrogen bonds, that's playing such an important role. All right, so we have DNA. We know DNA is double-stranded. We know it forms this double helix. Hydrogen bonds are important. They have these different nucleic nitrogenous bases. Let's look at RNA. So RNA is almost always going to be single-stranded. This sugar is ribose, not deoxyribose. All right, so again, it's just the change in the position of one single atom that causes the change of the entire molecule. Now the thing about RNA is that it uses uracil instead of thymine. All right, so it doesn't use thymine, it uses uracil. Now the reason we think it uses uracil is because uracil is energetically less expensive to create. Right, so we don't have to put as much energy to create this base compared to thymine. Right? And RNA doesn't have to be as stable as DNA, all right? And we have to make a ton of it. All right? So if we have to make a ton of it, we're going to use this less energetically expensive base. Now RNA is in several different forms that we'll get to later on in the course here. It can be messenger RNA, ribosomal RNA, transfer RNA, have many different functions here. All right? The key thing I want you to take away is that it's single-stranded, uses ribose, has uracil. All right, I kind of wanted to step back into kind of like the history of how DNA was found out because I think it's really fascinating and it's different than what I learned in high school and elementary school. So how was the secondary structure figured out? How did we know it was a double helix? All right, so it took a while, but chemists had worked out that the structure of those individual nucleotides. Right, they had worked out that structure. They also knew that DNA polymerized through those phosphodiester linkages, right? Now, Watson and Crick took this information and found out that, oh, they have a sugar phosphate backbone, all right? So we're building upon our information here. All right, a gentleman named Erwin Chargaff was able to establish 2 empirical rules for DNA, such that the total number of purines and pyrimidines are always going to be the same, all right, and that A equals T and G equals C. Now, based upon all this information, Watson and Crick was able to develop that, oh, it's a double helix, all right? They built upon the work of other scientists. Now, what we don't often hear is the work of this lady, all right, Rosalind Franklin. All right, so Rosalind Franklin and Maurice Wilkins, they had actually proposed this double helix structure of the DNA, had it almost in publication, and Watson and Crick got in there before them and published it, so they get all the credit. But what we don't hear is that they used Rosalind Franklin's image here, and this is an image she took of those molecules in DNA. Doesn't really make sense to me because I'm not a molecular biologist here, but they were able to look at this image, and this is what told them it was a double helix. Now, Rosalind Franklin did not give her permission to use this. All right, it was actually her partner that showed it to Watson and Crick, and then Watson and Crick used this without her permission to publish before her. All right, so she does not get the credit she deserves. Amazing lady that was able to discover the structure of DNA before Watson and Crick. So I think it's pretty interesting. I actually put an additional resource in there about her contribution. I really like this article because a lot of articles can be charged to really drive home one perspective, and that because she was a female, she deserves the credit, which, yes, is absolutely true. But sometimes people will exaggerate, and this article goes really into depth about literature that has been written about her that maybe exaggerated some principles, and maybe some other things like they were actually friends. So Watson, Crick, and Rosalind, they were actually friends. Now, she still didn't give him permission to use that image, but they were friends later on in life. So I think it's a really fascinating look into her life and the history of DNA. All right, so that was actually a really quick class period. I'll get you guys out of here a good 15 minutes early. There's also some videos to watch if you need help understanding protein folding. Prep for Friday, though. All right, I'm not going to have you watch a video, but you do have a couple assignments due. So one is the Metacognition 2 assignment. It's basically asking you to review how has your studying worked so far. And then the other one is Quiz 1. Now, for Quiz 1, I'm having you do it on Moodle. It can be open book. I just want you to get a feel for the type of questions I might ask, the level of depth of those questions I'm looking for. All right, so you're not so intimidated when we do other quizzes and when we get to the exam. All right, I'm going to make that available tonight. It's not on there yet. I promise I will put it on there tonight. As far as the H2O assignment, turn that in on your way out. Any questions I can answer? Yes?
Will all the quizzes be on Moodle? Or will we do some in class?
That I haven't decided.
Okay.
Yes.
Okay. And then if it's open tonight, sorry.
Oh, yeah.
Is it on the stuff that we've already talked about, or should I wait till like after Friday's lecture?
It's going to be on the stuff we've already talked about.
Okay, so I can take it like today or tomorrow.
Yeah.
Okay,
Yeah.
perfect.
I think it's due by Friday.
Okay, perfect. Thank you.
Friday at midnight. Yes. Oh, that is fine. Yeah.
So you want to do
I will put that on there.
Northeast Texas.
I'm so sorry. What city?
6.5.
Thank you.
Yeah, have a good day.
Crime per capita.
Thank you.
Yeah.